Thank you. তো আজকে আমরা অ্যাডাপটিভ ইমিউনিটি নিয়ে বলবো। প্রথম দিন আমি ইনেট ইমিউনিটি বলেছিলাম এবং এটা বলেছিলাম যে ইনেট ইমিউনিটি এন্ড অ্যাডাপটিভ ইমিউনিটি দে ওয়ার্ক হ্যান্ড ইন হ্যান্ড. So I'll go through the very basic concepts of adaptive immunity and tolerance. And so here is the concept that there are antigens, which is, and then antigens are recognized by the receptors. And then depending on how many times, how short or how long the interaction between antigen and the receptor occurs, the response is decided, and that is further modulated by the co-signals, which can be positive or negative or adherence, and that results in the production of different cytokines, IL-1 beta, IL-10, IL-12, TNF-alpha, etc. We had very nice presentations the other day. So basically, the amount of antigen, the form of antigen can decide the kind of MHC also they will engage, and these three together also decide some of the biophysical parameters of its interaction with the receptor, and these are affinity, avidity, and the number of receptors occupied for how long, short or long, or how many times repeatedly, and then along with the signals, positive, for example, through CD28, or negative, for example, through CTLA4, or adherence, for example, ICAM1, LFA1, we know that the effector function through different cytokines are decided. Now, this is the homeostatic set point at the very center of it. On one side, it is immunity, where we have the survival of the adaptive immune cells and their activation and their expansion or proliferation and their differentiation. Whereas on the opposite side, we have tolerance, which is inhibition or by death or by energy. Now, there are different molecules involved in this. For example, for survival, we need BTK, ITYK, or BCL2, BCLXL, and molecules that primarily delete these cells are the CD95. That means which plays a very significant role in the B cell and T cell selection, and then contextually promoting expansion or deletion of the TNF-alpha receptors, because they have dual functions, and so does have the CD40. So basically, what we have to understand that the homeostasis or immune homeostasis is maintained within a normal range by interactions between counteractive forces, counteractive principles, or counteractive factors, and these can be inherited or can be acquired. And accordingly, when some of the factors are predominant, we get immunity, and the other counteractive factors, when they are predominant, we get tolerance, inhibition, death, and energy. So in principle, once we know what are the factors that provide immunity to a pathogen, for example, then we can manipulate the whole system to derive the immunity against a pathogen. Alternatively, when we know that, okay, these are the immunity providing principles, we can block them, and then we can do the energy, induce the energy or induce death or inhibit them, and we can induce tolerance, and which is very important, for example, to make the allograft, accepted, we need to induce tolerance. So in that case, for example, we use immunosuppressants, which can be non-immunological or immunological. For example, we can have CTLA4 signaling heightened, and therefore we can induce tolerance, or we can induce PD-1 signaling and we can induce tolerance. So basically, what we need in principle to understand that there are certain factors which provide immunity, and there are countermeasures or counteractive factors which, if predominating, can induce tolerance. So immune homeostasis is at the middle within an acceptable range, where there are not natural interaction between these two different opposing sets of factors maintain this immune homeostasis. So next is, we know that the two major cells that are mediate adaptive immunity are the B lymphocytes, or the B cells, which are produced in bone marrow, and the T cells, which are produced in thymus. Now, the B cells develop in bone marrow, and they go through different steps like pro-B cells, early pre-B cells, late pre-B cells, then immature B cells, where we have the IgM coming on the surface. And then when they get matured, they also express IgT. And then when they're activated, they shed IgT. And then this IgM is switched to different subtypes or classes of immunoglobulins, for example, IgG, IgA, or IgE. And when these cells are activated and differentiated to produce different antibodies in secreted forms, then these cells become plasma cells and can be in bone marrow. So basically what is shown here, that different molecules and different events, for example, here you'll see the D to J, that means diversity genes to the joining genes, or variable region genes to DJ, diversified and joining genes, are combined in such a manner that we have a particular specificity immunoglobulin, or the immunoglobulin or the antibody with a particular antigenic specificity. Now, what is very important in this case here, you will see that there is a pre-signaling receptor here, M-shy LC, which means at this stage, the early pre-B cells to late pre-B cells, there is a checkpoint where the fate of the B cells are decided. And when these are joined by, again, another lambda or kappa, then they get another signal when they are again selected, and so that the mature B cells, when they are released into the circulation, and that they are now able to go to the site of infection or to the secondary lymphoid organs like spleen or lymph nodes, they have the very specific activity or recognition capability for the antigens, and then they can be activated and differentiated into plasma cells secreting the antibody, performing their functions. And then the T cells, as I said, they develop in thymus, and you will see that as they enter the thymus, the precursor thymocytes, or the precursors of T cells from bone marrow, they migrate through the medulla, and then they go through different stages like DN1, double negative 1, or double negative 2, or double negative 3, and then there is double negative 4 also, and then all these pre-double positive or the double positive cells, and then they go to single positive to become either CD4 or CD8 at the end. And this is the time when these mature, educated T cells leave the thymus through medullary sinus. Now, this is very important here, like the B cells. Here, when the T cell precursors come to the thymus, they have a potential to develop basically all sorts of T cell receptors that can identify just any antigen, even including autoantigens. Therefore, they have to be processed through different steps, like I've shown in the bottom, different double negative sets, to double positive, CD4, CD8, and then to single positive. And by the time they arrive, the single positive cells, CD4 or CD8, they undergo two processes. One is the negative selection and positive selection. And by this method, we ensure that we have the T cells which recognize only the foreign pathogens or the foreign antigens and not the self antigens. In the case that they are not educated appropriately, we can have these T cells which can recognize the self antigen and can destroy it. And in that case, we have the autoimmune diseases, for example, diabetes, for example, systemic lupus erythematosus, for example, Graves' disease, myasthenia gravis, and so many. So basically, what we find that for both B cells and T cells, selection is very important, and that is the part of adaptive immunity or qualifying this immunity as adaptive. So here again, I have shown the same, that in each step, we will see that there are quite a few factors which are responsible for the progression of double negative cells, DN1 through DN2A, 2B, DN3, DN4 to single positive cells. Now, we can, because during this process, we'll have both the negative selection and the positive selection. And many of these factors that are listed here play a very important role in cellular activation and differentiation. So any deficiencies or any exaggerated activities of any of these molecules may lead to impaired T cell education in the thymus. And in that case, we will have two different problems. In principle, one, we may lose those T cells with the receptors which can recognize these foreign pathogens or even transplant antigens, or we can have the cells which will have very strong self-reactivity. So basically, we can have these counteractive problems if we have a problem in this selection process because of any of the problems associated with these factors listed here. So here we have compared that you will see, because the T cell receptor also is a part of immunoglobulin superfamily. So they share the structure, and that is why everywhere you will see that here we have VDJ in case of B cell receptors at the top written in blue, that here VDJ recombination happens and then they expand and then again they give feedback. And as a result, here the first step where the selection happens, and then only those which survive and again expand, and then they go forward. And then we have the pre-B cells where you will see here VJ, kappa, and alpha. So basically, kappa and lambda. So basically, this is where we have again another round of selection by receptor editing, which is basically, we can say, a finite modulation of the antigenic specificities of these B cells, and then they mature to give us the immature B cells, which are IgM positive. Similarly, in case of T cells as well, we will have the pro-T cells, which are CD4 negative, CD8 negative. That means they are double negative. But then they first have the TCR beta gene rearrangement, VDJ beta. And then, like the B cells, they have this selection process here. Those which cannot survive or do not recognize the MHC class 2, for example, or class 1, the self-MHC, they are deleted. And therefore, those who survive, they get the signal, and then they have final expansion. And then they go to the pre-T cell stage, which is double positive cells, when we have the TCR alpha gene rearrangement. So we have to remember that the TCR beta gene rearrangement comes first, and then that is followed by TCR alpha. And when we have these two together, we have this immature alpha beta TCR positive, CD4 positive, or CD8 positive, single positive T cells. And these are the T cells then which can leave thymus. So basically, in all these processes, RAG1 and RAG2, that means recombination activation genes 1 and 2 coded products, are very important for mediating this process of education. So what happens? So basically, if we try to understand what is the importance of this selection process or this developmental process, which is adaptive, is the repertoire of newly formed B cells in bone marrow, or you can say repertoire of newly formed T cells in thymus. You will see it has a very, very wide specificity. And because of this specificity, what happens, you will see this red arrow here. So beyond this is all potentially autoreactive B cells or T cells. Now, during this developmental process, what happens? They are either deleted by death or their receptors are edited, and that actually gives the feedback, and therefore the autoreactive T cells are gone. So now, what we showed in the previous slide, what is that feedback mechanism? By that, what happens? This set point, trigger set point, is further left shifted. So you see here. So this is another round of selection, whereby you will see that only few cells on the left of this arrow are surviving here, and all these cells again they go out. So these are the cells which are appropriate cells and can form the germinal center. So what we find is the truly antigen specific safe reactivity are these cell populations. So basically what you can see here, that at the second stage, we have the repertoire of B cells or the T cells migrating to the peripheral lymphoid organs. And another round of selection there, we get the repertoire of B cells in circulation, which are safe. So basically, what we have tried to show here, that the reason we call it adaptive immunity is because it has that flexibility or the ability to adapt to a particular set of antigens or antigenic reactivity, and those have to be safe for the organism. And that is why we call here one is adaptive immunity, because this adaptivity is stemmed from the receptor rearrangement and also by receptor editing. So basically, this is what is in contrast with the innate immunity, where the antigen specificity is full and final, coded in the germline. So there is no gene rearrangement in case of innate immune cells. Whatever, for example, toll-like receptors on node-like receptors, that means NLR, or C-type lectin receptor, that is CLR, we have in those systems, they are not adaptive because they cannot rearrange their genes to fine-tune their antigenic specificity or to get rid of any kind of autoreactivity that would be harmful to the organisms. So this is basically the very concept of adaptive immunity, and what we know also of tolerance. So basically, this is again the repeat, that this is the first checkpoint for the B cells, where we have the pre-B cell receptor that gives some sort of signals, and then no pre-B cell receptors, those we have, so they undergo apoptosis. This signaling is very important for their survival, so that they can then expand and can express Ig alpha and beta chains, which is the B cell receptor, and then they can have, we can have the immature B cells with IgM. So basically, again here, no B cell receptor, they are undergoing apoptosis. So basically, what we can see here in case of B cells is also true for T cells, having two different checkpoints for positive and negative selection. So this is a very simple thing. I mean, I would like to simply describe that. Here we have the mu chain, which is coding for IgM, or delta for IgD, or gamma 3, which is G3, G1, G2A, G2B, C epsilon means IgE, and alpha. So what you will see here, that starting from early vertebrate, as we go up and up, what we are seeing is that the number of genes and their localization within the chromosome that have changed. And this is very important because this is what gives the flexibility or the fineness of the adaptive immune system that very finely tune the antigenic reactivity in mammals. So that is only concept that I would like to show from this slide. Now, when there is a problem of bringing, for example, this V to D and to J, like I showed in the previous slides, that we have this V, D, J recombination, along with all these things, others, that we get the final antibody. If we have a problem, then we don't have the antibody, or the antibody selected for only for an antigens, and then we have the autoimmune diseases. So basically, all those problems are hidden here. If this process of recombination and the switching is not performed appropriately, we have these diseases. So what is the importance of this is, you will see we have the inhuman variable chains kappa and lambda, light chains have 40 and 30, and heavy chains 40. So now you can imagine one of these 40 combined with any 30 of these, any 40 of these 8 chain gives us a huge recombination number. And now all those numbers are finally again multiplied by the diversity genes in the heavy chain, which is 25. So you can imagine here, if we have around 1200 or 1600, into each of these having combination probability of combining with any one of these 25, that means 1600 into 25, and then the joining of either 5 or 6 or 4 and 6, that means another 30. So you can imagine by combination of this V, D, and J, we can develop a huge number of antigenic specificity, and that is about 10 to the power 12. So basically, what happens, the mature B cells then express IgM and IgD as they are the first heavy chain segments in the locus. I have shown you earlier, mu and delta. And then what happens when the B cell is activated, then these D or all these portions are taken out. And then, for example, in case of IgE, now this VDJ is coming with epsilon chain. And then we have similar processes for all these. You see here AM, G3, G1, G2A, G2B, E, and A, all these different classes of 5 classes of immunoglobulin antibodies. So basically, that is what overall about the classes of antibody. But when we talk about the specificity, then this VDJ is very important, how those are changed. So this is here what you can, what I showed in the previous slide is further detail how from this continuous chain, there is a small nick here and there is a small nick on the left side. So now these two are combined together. So what happens, they go out, and then what we get is this VDJ portion. This is now linked with C gamma 2B. So basically you see here VDJ, then EMU, and then C gamma 2B. And when they are linked now together, this middle portion is excised out, and what we get is IgG2B antibody. So basically, this is the process which is very important. Now, this is called class switch recombination. Now, you see, if we don't have this AID or other cofactors that maintain this process or perform this process, then what will happen? Then we will not have this kind of class switching. And there is one very strong disease which we call X-linked hyper IgM syndrome, XIgM syndrome, where CD40 is not there or CD40 ligand is mutated so that CD40 signaling is not there. And in that case, we do not get this isotype switching. So no class switch. And what we get as a result, this IgM expressing B cells proliferate and they produce more of IgM, and none of them could be switched to either of these other classes. So we get hyper IgM syndrome. So basically, this is here again, it is important that to remember that this B cell selection happens not only in bone marrow, but also in spleen and lymph node. What happens if these B cells are self-reactive, then they cannot enter the germinal center, and then they do not develop into memory B cells and plasma cells. So basically, this is for the B cells. For T cells, we have even more elaborate system because the B cells can recognize unprocessed antigen, the antigen as is, whereas the T cell receptor cannot recognize any antigen as is. They do need processing of those antigens, big peptide antigen, polypeptide antigens, by different proteases into small peptides like 9 to 15 amino acid long for MHC class 2 combination, or 5 to 9 amino acids for MHC class 1 combination. So all this work is done by a group of cells which are called antigen presenting cells. Now these antigen presenting cells can be professional, which means they are always ready to acquire the antigen, process the antigen, and present the antigen to the T cells. The features of these antigen presenting cells are they express very high class 2 MHC. Class 1 MHC is expressed by all nucleated cells in our body. So they do also express class 1 MHC, and they do express very high number of co-stimulatory molecules. So basically that means these professional APCs have MHC class 2 and co-stimulatory molecules constitutively very highly expressed. And they secrete many different cytokines like IL-12, IL-18, TNF-alpha, IL-6, etc. And these cytokines provide the third signal. So the peptide bound to class 2 MHC or class 1 MHC provide the first signal when they bind to T cell receptor, and then the secondary signal or the co-stimulatory signal by CD28 when it binds to CD80 or CD86 on the T cells. And the third signal is through IL-12 or IL-18, which are secreted by antigen presenting cells and go to the T cells. So the fate of these interacting T cells is that these T cells are activated and later on they differentiate based on the cytokines environment. By contrast, we have a non-professional group of APCs which are low in MHC class 2 expression and low in co-stimulatory molecule expression. For example, neutrophil in case of human, for example, keratinocytes, for example, melanocytes, for example, glial cells, etc. So basically, those are not the readily active antigen-presenting cells, but if these antigen-presenting cells are placed in an inflammatory condition, for example, we have lots of TNF-alpha and interferon gamma, these non-professional APCs can behave or function like a professional APC. And in that case, they also secrete IL-12, IL-18, and IL-6, and the T cells are activated. Now, by contrast, we have another group of antigen-presenting cells, which are very low expression of or no expression of class 2 MHC or class 1 MHC will be there, and very little to no expression of costimulatory molecules, but they do secrete a lot of TGF-beta and interleukin-10. And this TGF-beta and IL-10 send signal through SMAD and IL-10 through STAT3 to energize these T cells, and these are called positively induced energy in the T cells. So they are the regulatory antigen presenting cells. So this is very important that these antigen presenting cells do not have the antigen specific receptor, in a sense that, okay, we have MHC which binds antigens, but then not like the TCR or BCR, but they do present the antigens to the T cells, which are important for not only T cell activation, but once the T helper cells are activated, they can secrete different cytokines to help the B cells to be activated, differentiated into plasma cells and secrete antibody. So these antigen presenting cells that way perform very significant functions. So basically here what I have tried to show is the binary logic of antigen presentation to the T cells. What we see here that these antigen presenting cells can capture from two different places, either endogenous or exogenous. Endogenous means, for example, when our cells are infected with virus, so they go to MHC class 1, whereas any other antigens taken off from outside, they are presented through MHC class 2. And then antigen processing, is it cytoplasmic versus endosomal, lysosomal? For example, when these antigens, endogenous antigens, they are processed through MHC class 1 cytoplasmically to go to MHC class 1, whereas exogenous go through this endosomal, lysosomal pathway to make the MHC class 2 peptide complex. And these MHC class 1 presented antigen are recognized by the CD8 positive T cells, whereas the MHC class 2 peptides are recognized by CD4 positive T cells or the helper T cells. So basically, this is what represents the binary logic of antigen processing and presentation to T cells. So this is where what we have shown, that this is where the endogenous antigen is coming out from the rough endoplasmic reticulum after synthesis, and then this is proteasomally degraded, and then this is transported, these smaller peptides are transported through this lumen, and with the help of tapacin and other molecule, they are where they are binding to MHC class 1 and calnexin, they are processed through Golgi complex, and then they are surfaced on the antigen presenting cells. Whereas you will see here, the exogenous antigens are engulfed and then they are processed through a phagolysosomal system, and then they are digested, and then they are actually the small peptides remove the cleave fragment from this MHC class 2, and they are complexed with MHC class 2, and then they are transported back to the cell membrane, where this MHC class 2 peptide complex is recognized by the T cell receptors on the CD4 positive T cells. Once the T cell receptor sees this or recognizes this peptide MHC complex, either MHC class 1 or MHC class 2, then all these signalosome complex forms. So you have different antigen, different enzymes or different signaling intermediates that activates RAS, RAF1, ERK1, 2, or through TLC gamma, all these phosphatidyl inositol phosphates, or diacylglycerol like DAD, or PKC theta in case of T cells, and all through these different signaling intermediates leads to T cell activation, proliferation, survival, differentiation, morphological changes, and cytokine gene expression. So it's a host of alterations in T cell functions. So basically, we have seen all this differentiation of these T cells, but what I found that in the previous talks, we had less discussion on the Tregs. So we have two types of Tregs, natural Tregs and induced Treg cells. So the natural Tregs are originated in the thymus, whereas induced Tregs are generated in periphery. And the phenotype, how they are recognized, both of them are FOXP3 positive, because this is the transcription factor which is required for acquiring the regulatory function of T cells. But the phenotype robustness is natural Treg is irreversible lineage, whereas the induced Treg can change their phenotype. So they're flexible or plastic. TCR stimulation, the natural Tregs are stimulated by self-antigens, whereas induced Tregs can be environmental. The TCR affinity is high to moderate in case of natural Treg, and induced Treg, it is suboptimal. And co-stimulation in case of natural Treg, it is CD28, whereas in case of induced Treg, it is primarily CTLA4. And the cytokines required for natural Tregs, it's not yet known, but most probably thymosin or thymopoietins are important. But in case of Tregs, induced Tregs, we need TGF-beta and interleukin 2. So basically, we have two different Treg populations, and this is very important, this induced Treg in the periphery, to fine-tune the amplitude and the quality of the T cell response in the adaptive immunity. So the interim summary is, what we have found so far is that B cells and T cells are selected in the bone marrow and thymus respectively for their antigenic reactivity. T cells help B cells produce antibodies of 5 different classes, IgM, IgD, etc. And then B cells class switch from IgM to IgE by IL-4, IgM to IgG by IL-4 and interferon gamma, and IgM to IgA by TGF-beta. And then B cells mature their affinity for antigens by VDJ recombination, somatic hypermutation, and nucleotide addition, all catalyzed by very specific enzymes. T cells also follow the same development and response. The difference is that the T cells do not produce antibody, but help antibody formation and immunoglobulin class switching by secreting different cytokines. And for T cell activation, antigen presenting cells are required for processing antigens and presenting the antigens to the T cells. And this is not required by the B cells. So now, with all this in the background, we have many questions, but I have chosen just 4 questions to exemplify what is the importance of having all these processes happening in the background. So the first question, what happens when the B cell selection or T cell selection goes wrong? So that is what we have found in the next one, that autoreactive B cells and T cells escape the deletion. These autoreactive cells circulate through the body, and the autoreactive cells attack the cell tissue, destroying those tissues, and such destruction of cell tissue leads to autoimmune diseases. Now you can actually characterize or associate these different autoimmune diseases to a particular type of immune response, and these are 6 different criteria that the autoantibodies or the autoreactive T cells with specificity for the affected organ are found in that disease. So in case of diabetes, so we can have the pancreatic antigens or even insulin, reactive antibodies or the T cells. Similarly, autoantibodies or the T cells are found at the site of tissue damage. So for example, in case of diabetes mellitus, we have this pancreatitis, and we can see those near these islets, all these infiltrating T cells or the B cells. The levels of autoantibody or T cell responses reflect disease activity, for example, in renal systemic lupus erythematosus or systemic vasculitis or antiglomerular basement diseases. And in many cases, we can see this. And then reduction of the autoimmune disease response leads to the improvement of the disease. So you can treat the patient with immunosuppressants, and then it will take some time to subside this disease because that suppresses the autoimmune response. Then transfer of antibodies to a second host, for example, mother to the fetus, transfer of IgG, or in case of bone marrow transplantation from an autoimmune donor, the donor is having autoimmune diseases, and if you take the bone marrow from that donor to a normal person, then he will have this autoimmune disease transferred, and then immunization with autoantigen induces an autoimmune response. So these are the different criteria by which we can actually associate an autoimmune disease to a particular response. Now, the second question was what happens if there is a defect in Ig class switching. So what happens? I have given one example here, X-linked hyper IgM syndrome. So in this case, it is caused by CD40 signaling deficiency due to CD40 ligand mutation or absence, affecting IgM switching to other immunoglobulin isotypes, like say. So because with that, these B cells which are expanding now are producing huge amount of IgM. So we have hyper IgM, and then the patients are susceptible to infections. The only treatment is bone marrow transplantation from a CD40 ligand sufficient donor. And the mechanism of the disease is that the B cells do not receive CD40 signals due to defective or absence of CD40 ligand. And in addition, the T cells are not functional to secrete B cell helping the cytokines. If TGF-beta is absent, then neutralization of the microbes by secretory IgA in the GI tract will be deficient, causing infections of the GI tract. So this is another example if we do not have appropriate immunoglobulin class switching. So defective affinity maturation, what will happen? Like I said at the very beginning, why we call it an adaptive immune response? That's because we adapt to the requirement of the immune system or the organism. So what happens? This affinity maturation actually represents that adaptability. So a hallmark of antibody response is affinity maturation of immunoglobulins. This affinity maturation works as the directed selection of B cells producing mature antibodies in lymph nodes and spleen. The affinity maturation is caused by somatic hypermutation so that the antibodies acquire higher affinity during the course of an immune response, and affinity maturation is therefore crucial for neutralizing or fixing the pathogen for killing more efficiently. And impaired affinity maturation is therefore not a complete defect in antibody production, but is related to the strength of antimicrobial functions. So here, what happens to the defects in adaptive immune response to infection and tumor? This itself is actually several lectures. So I have just pointed out in one slide some sort of summary that defects in adaptive immune response come up from escape from deletion causing self-reactivity, holes in the repertoire, that means covering fewer antigens. This is the major problem. And then I have discussed already self-reactivity in autoimmune diseases, and holes in the repertoire covering fewer antigens means that increases the susceptibility to infections, increases higher risk, heighten the risk to tumor development, and heighten the suppressive T cell functions. But does it mean better recipients in case of transplantation because they are compromised? So if we have this organ transplantation versus bone marrow transplantation, what we find is that whether the host cytoablation is required, yes, in case of bone marrow transplantation, but not necessarily in organ transplantation. HLA matching is essential, very critical for bone marrow transplantation. Chief complication, what happens in organ transplantation is rejection. But in case of bone marrow transplantation, because these will be the different leukocytes coming out from these bone marrow cells, so we'll have graft versus host disease, which we call GVH, and whether they are treated to be immunosuppressions. So this is uncommon, but here we have to treat it. And then terms for success, it is acceptance when we call it success, And in case of bone marrow transplantation, we have to induce tolerance, and that is the term for success. So the take-home message for this adaptive immune response is that the cells of the adaptive immune system include antigen-specific receptor-bearing T cells and B cells. B cells do not need a processed antigen for recognition. T cells see only processed antigens on the antigen-presenting cells when presented in the context of MHC class 1 or MHC class 2. And APCs can be professional or non-professional or regulatory. And these defects in antigen presentation leads to deficient immune response. Defects in B cell selection may lead to escape of autoimmune B cells, causing autoimmune disorders. Defects in T cell selection may lead to escape of autoreactive T cells. Deficient B cell functions may include different immunodeficiencies, primary immunodeficiencies, someone will teach it, and then deficient T cell functions may include different immunodeficiencies. So when the primary immunodeficiencies will be discussed at that time, all these things will be discussed in detail. And these deficiencies differentially affect responses to pathogens, tumors, self-antigens, and transplanted tissue. Now, before I complete this, I would like to say that based on this, one can see that there are very broadly categorized into two kinds of responses. One, which is directed against pathogens and tumors, which are pro-inflammatory. And in case of immunity, for example, the self-antigens or transplanted tissue, we need anti-inflammatory cytokines. So for the first two, pathogen and tumor, we need pro-inflammatory cytokines and the processes which will lead to those pro-inflammatory cytokines, whereas self-antigens, to suppress the autoimmune diseases or to make the transplant accepted, we need anti-inflammatory cytokines like TGF-beta and IL-10. So thank you very much for your kind attention.